And now I'd like to hand the screen over to Doctors Patrick Mulholland and Jennifer Rinker. Patrick and Jennifer are part of the strong drug addiction neuroscience community at the Medical University of South Carolina with NIAAA and NIDA funded research centers and T32 training grants.
Thank you, Nafi. So today I'm just going to discuss what we've done so far using TDT fiber photometry system, and I'm going to provide a brief overview of where the field currently is in addiction neuroscience, and just give you an idea of what we study and why a fiber photometry system was useful for our work. So for those of you on the call that aren't familiar with addiction, I just wanted to provide a brief overview of some of the hot areas of interest in neuroscience field right now, and again highlight some of our recent work on plasticity and genetics that led us to incorporate fiber photometry. So recently, the American Society of Addiction Medicine updated its definition of addiction to reflect what the neuroscience field has discovered over the last 10 or 15 years. As you can see, they've defined addiction as a primary chronic disease of brain reward, motivation, memory, and related circuitry. It's also characterized by an inability to consistently abstain, impair, behavioral control, craving, and so on and so forth. We also know that genetic factors account for about half of the likelihood or risk that an individual will develop addiction. So, based on this, we're interested in three key areas. The first is circuitry, the second is behavioral control, and the third is genetic factors. As I said, right now the addiction field is intently focused on understanding the role of neurocircuits in the addiction process, and some of the key circuits are highlighted here in this slide. It shows that the circuits and the different neurotransmitters involved in different aspects of the addiction process. We are particularly interested in the prefrontal projections down to the accumbens shell, and again we're interested in the PFC because of its control over behaviors and cognitive processes. In addition to our interest in the circuits, we've learned a lot over the last few years about how chronic alcohol affects function of the prefrontal cortex in mice and monkeys. So our group at MUSC has shown that chronic alcohol exposure affects spike timing dependent synaptic plasticity. It also affects morphological plasticity, and it can influence how neurons in the prefrontal cortex fire. We think all of these functional and morphological adaptations are critical for the cognitive deficits that are observed in alcoholics, and we think that it's critical for the impaired behavioral control over alcohol-seeking behaviors that we know exist. So in addition to our interest in functional and morphological plasticity, we also study potassium channels because they are potent regulators of excitability, many different aspects of excitability and synaptic plasticity. And what sparked our interest recently was a study showing that Kv7 potassium channels in the prefrontal cortex can control how long an animal will drink a rewarding solution. This was interesting to us because we know that alcohol-dependent animals will drink more alcohol, and when they do drink, they drink in longer bouts. So we've spent a lot of time exploring the link between potassium channels and alcohol, and we started with some genetic analyses. And what we found is that there are some potassium channels in the prefrontal cortex where their expression levels negatively correlate with alcohol consumption. And this suggested to us that if you can increase the activity of potassium channels, you may reduce alcohol drinking. So these data that I'm showing here in the correlation are from BXD recombinant inbred strains of mice that are genetically diverse. But we also know that in addition to these data, that there are SNPs and KCNQ genes in mice and humans that are linked to alcohol intake. So this became a pharmacogenetic target for us. When we administer Kv7 channel positive modulators to mice with a high drinking phenotype, we see that it nicely reduced consumption, especially at the early time points. So based on all of these preliminary studies that we've published recently, we wanted to get into the analysis of circuitry while the animals are performing tasks. So we saw huge benefits to employing fiber photometry into our research program. So these are just two examples of how photometry has benefited us so far. It allows us to determine how excessive drinking and alcohol dependence alters circuitries, in particular PFC projections to the accumbens, and how Kv7 channel positive modulators that reduce alcohol intake, how they can influence activity in subpopulations of neurons or even specific circuits during ongoing behaviors such as alcohol drinking. So now I'm going to turn it over to Jen Rinker, who has spearheaded all of the photometry studies in our lab, and she's going to describe to you the data that we've collected to date on alcohol intake and GCAMP6 signaling in PFC neurons. Thanks.
Okay, let me get in the driver's seat here. Okay, hopefully everybody can see this. So as Patrick said, we are particularly interested in the cortical involvement in voluntary ethanol consumption because the medial PFC is so critically involved in regulating subjective effects of drugs of abuse and responsivity to drug-associated cues and reinstatement of drug seeking. So we really wanted to try to understand the role of the medial PFC outputs in voluntary home cage drinking devoid of cues or novel drug-associated contexts. There's a little bit of data out there from in vivo ePhys suggesting that inhibition of the medial PFC is crucial for control over consumption of a rewarding sucrose solution. So Horst and Laubach showed this in their 2013 paper, demonstrating that the event-related spectral power in the prelimbic PFC is decreased during the consumatory phase of consumption of a sucrose solution. This was not related to just merely licking behavior. As you can see now here in this bottom graph, the animals were allowed to lick from the sipper tubes prior to the reward delivery through the sipper tube. And so this demonstrated that the activity was really, the decrease in activity in the PFC was related to the consumption of the reinforcing solution. Barker et al. Also recently showed that the infralimbic cortex of the PFC shows a similar pattern of behavior. So there's an increase in firing rate just prior to consumption of the reinforcer sucrose solution, which the firing rate decreases during the consumatory phase. So the goals of the current studies that I'm going to present to you are kind of twofold. And one of the drawbacks to in vivo ePhys is the lack of cell type specificity. So in both of these studies, signals could be reflecting pyramidal projection cell activity as well as interneuron activity. And we wanted to determine if these signals were being driven by specific cell subtypes and if the response would be similar when an animal was consuming ethanol. And so we hypothesized that medial PFC pyramidal projection neurons would show a similar pattern of activity, so a decrease during the consumatory phase that would scale with the value of the reinforcer. Additionally, we wanted to determine whether ethanol dependence altered calcium signaling in response to ethanol consumption. So this is just a general overview of the studies that I'm going to present to you. We use C57 black 6J mice from Jackson Labs. And for those of you who are not familiar, we choose this strain because they're an ethanol preferring strain of mice and they readily drink ethanol solutions to the point of intoxication. We achieved cell type specificity by infecting the prelimbic PFC with an AAV1 with a CAMK2 promoter to preferentially infect putative glutamatergic projection neurons. There are a lot of flavors of GCAMPs out there, and at the time, GCAMP6F was available through Adgene. And so we chose this variety because of its good signal-to-noise ratio and its enhanced decay kinetics. Mice were simultaneously implanted with a 400 micron multimodal fiber optic probe, which we made in-house with Thor Lab parts. And after about 5 weeks post-surgery to allow for transduction and stable GCAMP6 expression and responsivity to our positive control stimulus, which is generally a puff of air or a brief tail pinch, which causes a sharp increase in medial PFC calcium activity. We then began testing the animals. So here is just a sort of diagram showing you approximate probe placement in the prelimbic cortex and GCAMP6F expression, which was largely restricted to the prelimbic subdivision. So here is our TDT system, sort of cartoon, connected to our mouse in their home cage environment. And this allows us to do recordings during naturalistic behavior and allows us to see calcium-dependent changes in activity independent of any novelty or context-induced changes. To validate our setup, we placed the animal under brief isoflurane anesthesia and correspondingly saw a sharp decrease in our GCAMP6 signal, which you can see here. And there was not a corresponding decrease or there was no change during the isoflurane anesthesia in our 405 calcium independent signal, which is represented here on the bottom in this trace. And this was true across all 5 mice that we tested in this particular cohort. As you can see here in this heat map, during isoflurane anesthesia, there's a sharp decrease in GCAMP calcium-dependent activity. So just a little bit of data analysis housekeeping. There are a number of different ways to post-process your raw calcium signal from the TDT system to get your change in fluorescence or percent delta F over F. So the current method that we are using to process is kind of shown here in these images. So in this first image, you have the, in the blue trace, you see our 490, which is our calcium-dependent GCAMP signal. And then in the purple, you see the 405 calcium-independent signal. These 2 signals are normalized independent of each other by fitting each signal against a polynomial curve fitting function against time to generate our normalized signal, which is shown here. So the 490 normalized signal is now in green, and the 405 normalized signal is now in gold. And what you can see is that this detrends both the data sets, which show a slight drift due to photo bleaching. The 405 signal is then subtracted from the 490 to generate our final delta F over F calcium dependent signal, shown here in this final graph in green. So back to our experimental setup. So we have the animals again in their home cages, and they're given access to a fluid reinforcer. So I'm going to show you data from water, ethanol, and sucrose. Their home cage is set up with a grid floor so that we can connect them to a leukometry circuitry, which is run through MedPC, which outputs a TTL to our TDT system, allowing for us to time lock licking behavior to our calcium signal. So animals are given access to one of 3 solutions, so either tap water, 20% ethanol, or 1% sucrose solution, using the drinking in the dark model. And so this model, for those who are unfamiliar, just briefly, 3 hours into the animal's dark cycle, they get 2 hours access to the fluid reinforcer. And this is repeated across 4 days. And on the 4th day, we do our fiber photometry test, where we record calcium transients across that 2-hour access period. So week 1, they got 4 days of water, and on the 4th day, we recorded. During the 2nd week, they got access to 20% ethanol, and then on the 3rd, 1% sucrose. So next, I'm just going to show you a little cartoon of our calcium activity output to orient you to the video that I'm about to show you. So here on the y-axis, you're going to see the percent delta F over F. This is our change in our GCamp signal, which is represented by the green line. Across the x-axis is time in seconds, and the orange tick marks represent licks for, in the video it will be ethanol, but it also corresponds to licks for water or sucrose, depending on the reinforcer available. Okay. So you'll see that the animal is going to start out down here in this lower left corner. The spout, which contains 20% ethanol, is in the upper left, and you'll see the GCamp signal tracking across time in the top half of this panel. So you'll note a peak before the animal starts the licking bout, which dramatically decreases during the bout for ethanol. And you'll see several times he stops licking and the signal goes back up, and if he starts again, it goes back down. And this repeats across several bouts of licking for ethanol. And I'll show you here in a second, he's going to inspect the sipper tube, which is an empty port, and you can see the signal goes up, and then he goes back over to drink for ethanol. So here I'm showing in this top trace, this is calcium signal in blue now for water. And again, the tick marks represent licking for water, so you can see the bouts for water here in this first trace. Green is bouts for ethanol, and along the bottom in red is bouts for sucrose. And one thing I would like to note about these three signals is that the animals only ever consume the reinforcer available during extended periods of increased calcium activity. So you can see that the signal here is up, and the animal has a bout for water, same with alcohol, and same here with sucrose. But the interesting thing is that this extended period of activity is elevated for ethanol compared to water, and even more so for sucrose compared to ethanol. So there's a number of different ways that we can analyze this data. So just back to our little cartoon for a second. We are going to average the calcium signal across bouts across animals. So in order to do this, we take the 30 seconds preceding the initiation of a bout and about 2.5 seconds into each bout to look at the signal prior to initiation, and then the same thing on the tail end, about the last 2 seconds of licking and the 30 seconds post-bout. And when we average these signals across animals and across bouts, these are the average traces showing the standard error of the mean in the area filled in around the average, which is the dark line in the middle. And we have water across the bottom in blue, ethanol here in green, and sucrose in red. And what you'll notice is that there's a ramping in the calcium activity in the prelimbic PFC just prior to the initiation of a bout for both ethanol and sucrose, which is a little bit more elevated for sucrose, especially compared to water. Again, I said there was a number of different ways we can analyze this data. So going back to our cartoon, we also looked at the 60 seconds preceding the bout just to look at the sort of extended period of elevated activity prior to the initiation of the bout. We look at the signal at the onset of the bout, at the valley during the consumatory phase, and after the consumption. We also looked at the slope of the change in the signal from onset to offset, just as another way to sort of analyze the decrease in signal across the consumatory phase. So if you look at the 60 seconds prior to bout initiation, averaged across all of our animals, in 10-second bins, you'll see that the signal is significantly elevated for sucrose, especially compared to ethanol and water. And so we kind of interpreted this as the signal is scaling with the presumed reinforcer value. Signal at bout initiation was much higher for both ethanol and sucrose compared to water, and only slightly higher after the termination of the bout for sucrose. The change in the slope during the bouts was much greater for ethanol and sucrose compared to water, And if you look at the change, so over here on the left, I'm showing the slope calculations for the very first bout for water on the top, ethanol in the middle, and sucrose on the bottom, compared to their last bout during a session over here on the right panel. And what you can see is that across or within a session, the degree of change between initiation of the bout and termination dramatically decreases for ethanol and sucrose, but doesn't really change for water. And so we interpret this as an indication of satiety or diminished reinforcer value across a session. So just to sum up that data, we showed that calcium signaling in glutamatergic medial PFC neurons ramps up prior to the initiation of consumption of a reinforcer, particularly ethanol and sucrose, and this scales with the presumed reinforcer value. Inhibition of medial PFC activity is sustained during bouts of licking, and the degree of inhibition, again, is reinforcer dependent. And again, across a drinking session, there is a diminishing of the signal prior to the initiation of a bout, which may be indicative of satiety and diminished reinforcer value. So now I'm going to talk about the second goal of some of these studies, and this was to determine if ethanol dependence alters calcium signal dynamics in the medial PFC during ethanol consumption only. So we're no longer going to be looking at water or sucrose, but just ethanol. So basic study design, animals had 3 weeks of baseline drinking in the drinking in the dark style that I presented before, where they got 1 bottle access 20% ethanol. Then they had 4 back-to-back cycles of chronic intermittent ethanol vapor exposure. And this is where they spend 16 hours a day in vapor chambers, ethanol vapor chambers, and 8 hours of withdrawal. And this happens 4 days a week. And then they have 72 hours of withdrawal between each cycle. And then we allow them to go back into their home cages and drink the 20% ethanol solution, and we record during their test day, again, so day 4 of the drinking in the dark. So our results show that compared to baseline, the animals that went through vapor chamber exposure here in the red show a significant escalation in their ethanol consumption, which is indicative of dependence. Compared both to the air animals who did not go in vapor chambers and compared to their own baseline. And again, this is also reflected in the number of licks per 2-hour session. And so when we look at those average traces, again, here's the little cartoon up here in the corner just to remind you what I'm analyzing. We see this is for the air animals. So these are the animals, our control groups. And this top light blue trace is their initial recording for ethanol consumption. So you see the same trend, ramping of the signal just prior to initiation, a drop-off during the consumatory phase, which rebounds after. And what you see is that after 4 weeks of air vapor, so they just kind of sat in their home cages for 4 weeks, you see a diminished activity, a calcium-dependent activity in the medial PFC during, leading up to, during, and after consumption of ethanol, which may be indicative of diminished reinforcing value. The animals that went through ethanol vapor chamber exposure, on the other hand, did not show this same diminishment, and in fact, they even showed a slight enhancement at the peak just prior to the initiation of ethanol consumption and a slightly stronger valley during the consumatory phase. When we average this data, looking at the peak before the bout, during, and after for the control animals here on the left and the CIE animals here on the right, you see that there's a diminished calcium signal in the air animals from their initial drinking test to their post-air drinking test, which is not reflected in the dependent animals. If you quantify this in another way, so we're looking at the change in percent delta F over F from, so basically the difference from here to here and here to here. You can see that there's no real change in the air animals, but it's slightly greater. So this, if you look at, sorry, it's from here to here and then here to here. the degree of the change is no different pre to post for the air animals, but for the dependent animals, you see a slightly greater increase in the decline during the consumatory phase. So just to sum up that data, we've shown that calcium signaling, again, peaks prior to bout initiation and is maintained and are maintained in dependent mice and is slightly diminished in non-dependent mice during ethanol consumption. Frequency of events, and I didn't show this data for the sake of time, does not change, but the peak signal significantly decreases in the air mice, which is consistent with that average trace that I showed you. And this last little bit of data that I'm going to show you is also from a group of mice that were ethanol dependent, so they went through the chronic intermittent ethanol vapor exposure. And we administered a Kv7 channel opener, which Patrick talked about before. So this drug, this is just to demonstrate that you can see drug-induced changes in calcium activity in using fiber photometry. And so here we have two different mice, and this is a calcium trace after vehicle administration. And then you can see a dramatic change in sort of this oscillatory behavior of the calcium signal after they're given a Kv7 channel opener. And that is all I have. And so just some acknowledgement of all the people that and funding that went into all of this work. And then I think we're ready for questions.
All right, thank you very much, Dr. Rinker. I see the first question coming in here for you is asked from Nolan, and he's asking, just curious, have you empirically tested or compared signals at different time points post-viral injection?
Yes, we have. So that's part of why we wait 5 weeks, and this is sort of testing that we've done across a number of different cohorts of animals. The signal tends to be greatest about 2 to 3 weeks post-infection. At about 5 weeks, we get stable GCAMP expression. There may be a slight decline over the course. So all the data that I showed you today, these animals were tested over the course of about 16 weeks. There may be a small decline in the raw signal that we get from the GCAMP. But again, everything is normalized within sessions, so I'm not sure that that's a huge issue. If that answers the question.
Okay. Yeah, Nolan, if you have any, oh, yes, he says thank you. Okay, then. And Matthew Carter asks, can you please talk more about how you converted the raw signal trace into the final delta F over F for publication? Did you use a custom Matlab algorithm?
Yes, so we did use a custom Matlab algorithm. It was sort of pieced together from a number of different papers that have already been published, and in consultation with Doric lenses per their recommendation for how to analyze the GCAMP signal.
We're happy to share the code with anybody. If you reach out to us, we can email you what we have on it.
All right. And Matthew says thank you. So let's see the next question, and I think all of us can pitch in on this one, is how does one determine the optimal modulation frequencies with a locket amplifier? If Jen and Patrick, if you don't mind, I'll go ahead and start with the answer, and please feel free to add in anything you'd like to. So basically, if with the TDT software, it starts by default, channel 1 is 210, channel 2 is 320, it keeps going up from there. One thing you'll notice of those default frequencies is that they are neither harmonics of each other nor harmonics of 50 or 60 hertz. So basically, with the lock-in amplification, of course, it's only going to pick out the frequency that that you set it to, and it does it with a very large amount of precision. If you vary by more than a hertz or so, it is almost completely shunted out. However, of course, anytime you're generating any signal of a certain frequency, you'll get what's known as harmonics at multiples of that frequency. So one possible source of contamination is your lights above, which, depending on if you're in North America or in Europe and Asia, will either be modulated at 50 or 60 hertz, or if you're using incandescent bulbs, it'll be twice that since it's power, so 100 or 120. So that'll be light that's actually flickering on and off and could leak into your sensor. However, you'll also have a cable going from your sensor to the preamplifier, so that cable will be subject to picking up electromagnetic noise. So you want to, which would then be picked up as an optical signal by the system. So as a result, you don't want to choose anything that's at 50 or 60 or a harmonic of it. And when choosing your different light sources, you want to make sure they're not harmonics of each other. Other than that, pretty much any frequencies will work. However, if you do see any noise signals that aren't accounted for, first thing that I would do is try varying those frequencies just to make sure that there's not some electromagnetic source that's interfering. Okay. And then I see a question here is asked earlier for me from Sophie. She says that she has a basic question regarding the setup. And if I could explain why I advised against using different LEDs for each mouse and not to split the signals. So the thing is, when you have a fiber, if it's a simple linear fiber, the light really only has one source to go. So it just travels from the entry of the fiber to the output, and so you're just going to get a minimal amount of signal loss just because of from absorption of the light from the material, which is going to be minuscule. Anytime you bifurcate that cable and you start giving the light multiple light source pass, basically it's going to take all possible paths. So as a result, once you bifurcate that cable, you're going to be splitting half the power. Also, the light's going to be bouncing around inside the fiber now, and so not only are you going to get greater signal loss, but you're also going to you can't guarantee that the division down the bifurcated cable is going to be a perfect 50-50. So now if you're trying to basically, and of course, to minimize bleaching, but to maximize signal to noise ratio, you're going to optimize the amplitude of the light that you're putting out so that it's as high as possible to get the signal to noise that you need, but as low as possible so your bleaching is little as possible. If you're now not getting equal power between your two animals, now you've got less range and you've got more variability in basically how much power you're sending down to each animal, and so it makes it very difficult to optimize your recordings. You'll notice that when I mentioned about the G camp and the R camp, if you're using those two, you have to use a split cable to combine the signal into a sensor. However, at that point, it's not as dramatic because we have such a high signal to noise ratio because we're using the lock-in amplification, that even with that signal loss, you should still be able to get an ample signal. Okay. And here's another question addressed to all the speakers. Do you have any experience with homemade implantable fibers, or do you order from Doric or another company? If so, is it possible to reuse fibers? And since Jen and Patrick, since you guys have much more firsthand experience than me, would you like to start off the answer?
Sure. We have ordered implantable fibers through Doric. We also make our own in-house. Obviously, there's you have to do a cost-benefit analysis in terms of the price of the Doric fibers. I think are like 20-ish per implant, maybe.
I think the price just increased, actually.
Increased, okay. As opposed to the time you spend polishing the ferrules and the fibers in-house. In terms of reusing them, I would say no. We secure the fibers on the skull of the animals using dental cement or dental resin. And when you remove that, there's really not an easy way to get that off of the skull without damaging the fiber. Not to mention that implantation itself into the brain tissue is probably going to introduce some damage to the fiber tip. And so I wouldn't recommend reusing them.
Our in-house fibers cost around $10 and maybe an hour or so to polish. So you'll have to see what your budget is and compare that to commercial costs.
And I know that one advantage of the Doric fibers is that they had actually worked at the suggestion of Tom Davidson. Something he mentioned that was a common problem is that they basically worked on minimizing the bleaching problem. So in general, the Doric fibers will bleach more slowly than other fibers available. I also, too, as I recall, he mentioned to me that Thor Labs, they have a kind of a quick disconnect clip that he's been using. So I believe that allows you to connect fiber without having to cement it to the animal's head. Problem is, so that clip, you know, it does add weight to the animal's head, so it's not going to work well for everyone's recordings.
Yeah, just to clarify, we use the red clips, but that connects the patch cord coming from the TDT system to the fiber implant that's on the animal's head. But I'm not aware of a way to temporarily implant the fiber. I think the fiber has to be a permanent implant. So once that's removed, I don't think you can reuse that.
Ah, okay. All right, that's good for me to know too. All right. I hope that answers your question, Johannes. So next question is, how do you deal with different background fluorescent signals between fibers when comparing between days animals, as this can alter the delta F over F calculation?
So I think the best answer to this is that, you know, so we're talking about within one animal, is that what the question is? Using different patch cords and whatnot, and looking at different noise, background noise?
So that, if David, if you have a, yes, he says yes within animal.
Okay, so within the animal, signal change, because everything is normalized within session to that 405, and it's normalized to the GCamp signal within that session, I would say that there really isn't a huge issue across time or across different sessions of recording. I typically use the same patch cable and the same photometer and all that stuff for each animal for each recording, and I haven't had any issues with signal changing or background noise changing, if that answers that question. Patrick, did you have any? No. So everything is normalized within session to that animal's signal. So it's all the percent delta F over F is a change in the fluorescence based on what that animal's baseline is. So by normalizing it, you should take care of any across session differences.
All right. And then next, Vivinian Chi, I hope I'm pronouncing your name properly, asked, have you also generated stimulus pulses from the laser from the processor as used for optogenetics? So yes, that is actually, I hope you guys don't mind if I answer. Basically, yes, that is a very common application. That's something that can actually easily be done with the system. Since your optogenetic stimulation is not going to be, you know, sinusoidally modulated like the other signals, it will be filtered out. However, you do still need to optically filter the optogenetic stimuli. Obviously, it's going to be very bright and would saturate the sensor. So if you're building your own kind of breadboard series of cubes, you just need the appropriate filters so that the light for your optogenetic stimulation is not going to the sensor. And if you're using the Doric mini cube, they actually have a few cubes that are specifically for optogenetic stimulation, so that you can do those concurrently. All right, next question is from Mark Basista. It says, what is the shortest time scale of fiber photometry data do you work with? Do you ever attempt to look for single action potential? Yeah, so single APs, I assume action potentials for different length frequencies of bursts.
Well, the calcium signal is going to be much slower than a single action potential. And remember that what you're recording is really comparable to probably a field of population activity rather than action potentials from individual neurons. Sum of activity across all of the neurons that are underneath the fiber optic that's implanted in the region. So we are not examining millisecond time frames in our photometry recordings. The events that we see typically occur at a duration of 1 to 10 seconds. And Nafi, if you share Jen's screen, is Jen's screen still?
Yes, your computer is still the presenter.
Yeah, so you can see in those graphs that the action potentials, the GCamp signal does not necessarily follow the action potentials. The bottom right graph shows that clearly, and it also shows a long time scale of decay. So everything that we analyze is on a second scale, and that's typical for what I've seen in the published literature.
Okay, yeah. And if I may add to that, one of the advantages, both the advantages and the disadvantages of fiber photometry compared to electrophysiology, is that it is a more generalized recording. So that's a positive thing if you're looking at a larger population of cells. But of course, that means that it's not suitable for single units. Okay, next question is from Ritu Chowdhury. Again, I hope I'm pronouncing that correctly. What is the recommended distance between injection site and fiber site?
I generally implant my fiber about 10 microns dorsal to the injection site. And that's completely arbitrary. It's just what's worked for me. I'm sure you could ask other people and they have other recommendations, but that's what we've been doing.
Also, Patrick Hollows, he made an excellent point on the note on frequencies for lock and demodulation, that photo receivers have a limit of bandwidth, like the Newport 2151, with, and by default, their setup is maxed out at 750 hertz. And yeah, that is important consideration. They do have a low pass filter in them, so yeah, you cannot modulate above 750 without losing your signal. Okay. And then next question is from David Kupferschmidt. What are some pitfalls of subtracting 405 nanometer derived signal from the 490 nanometer derived signal? I've heard of examples where the 405 signal reduces when the 490 is elevated. Thank you.
That's true. The 405 is not a true isobestic signal. If you look into the literature, there is a bit of calcium dependent signaling that occurs when you stimulate with a 405, and it actually goes in the opposite direction of the signal that you get with a 470 or 490 LED. So we've struggled with this a little bit on what is the best way to control for photo bleaching and to control for movement artifacts. And while the 405 is not perfect, I think it is the best option that's available right now as a internal positive control. The other thing to consider is that the 405 signal may photo bleach at a different rate than the 470 or the 490 signal, and the analysis that we're currently, the analytical approach that we're currently using takes into account possibilities and differences in photo bleaching rates across those two channels. So I don't have a great answer, but again, I think the 405 as an internal control is the best option at this point.
Great, thank you. Yes, thanks. Okay. All right, next question is, we have a NeuroPort photo receiver 2151. What configuration should that be in when running an experiment in terms of gain setting switch? And for those unfamiliar with the 2151, it has 3 settings: DC low, AC low, and AC high. And he says, how does changing these affect your results? Maybe I'll go ahead and answer, and Jen and Patrick can add in if they have any thing they'd like to say. Generally recommend using the DC low. The problem so the DC and the versus AC, what that's referring to is DC versus AC coupling. So the DC coupling it's a direct connection with no high pass filter, whereas AC coupling has a capacitor in series, so it's actually removing any DC offsets. The problem with removing DC offsets is that if you have a DC offset, which pretty much any amplifier will, since that doesn't get removed, you have no idea how close you are to the saturation limit on the sensor. So as a result, you can end up saturating, and the TDT software has a built-in saturation detector to let you know, but it's based basically on the voltage of the signal that's coming in. So if you have it as AC coupled, you can easily be saturated. The system will not know, and you might be left scratching your head wondering what happened to your signal and why you're not seeing any GCamp response. So for that reason, we generally recommend using the DC low. And particularly too, you'll oftentimes find that especially the AC high, the high versus low being high gain or low gain, the high is you're very likely to end up saturating your sensor. And so basically that's the main things that affect changing the results. If you do find that you're not saturating and you're trying to record a really, really tiny signal and would benefit from a higher gain, then it is possible to use AC high, but what you're going to want to do in that case is you're going to want to keep a very close eye on the raw signal. And so basically on the TDT system, the default is called FI1R, and there'll be a number of channels equal to the number of drivers you're using, as well as the number of sensors. So for instance, if you're using 2 drivers and a single sensor, the first 2 will be the raw output on the for channels 1 and 2, and the third will be the raw input on the sensor. And so you just want to keep it, you'll need to keep an eye on that, on the raw signal, to make sure that it's not plateauing, to make sure that you're not saturating.
The only thing I'd like to add to what Nafi said is that TDT tech support can patch into your system remotely while you have an animal connected to the system, and they can teach you within minutes how to determine if your signal is saturated and why DC low is the best option to use. And we learned that within probably 5 minutes of Nafi looking at our recordings while he was in Florida and we were in Charleston.
Yeah, thank you, Patrick. Okay, we're starting to run out of time. We're actually going over, but if you guys don't mind, I would like to try and tackle a few more questions. So Ritu asked what volume of brain we are measuring from. Would you like to start? He's asking what volume of the brain are we measuring from. I believe you mean, well, what is a typical volume do you get a signal from in a typical setup?
So it's the I guess field is about 400 microns wide by 300 or so microns deep is about how the level of penetration below the fiber tip that you're recording from.
And it's 400 microns wide because we use 400 micron fibers for our recordings. Some people use smaller diameter fibers, so your field would be reduced.
All right. Next question is, what is the LED power to excite Gcamp as measured from the end of the patch cord?
So this has changed for us dramatically. So originally we were using the Thor Labs dichroic mirror setup that Nafi showed in the beginning. And with that setup, our output at the patch cord was about 30 microwatts. Is that right, what we were shooting for? And now we've switched to the Doric mini cubes, and with those, we only have to use 3 microwatts of output power.
Anything higher than that causes clipping in the system in our current configuration.
Okay. All right, I'm going to start trying to go through these. We still have quite a few. I'm going to try to address as many as we can. Oner has a quick, he's mentioning that he reuses the fibers from Doric and cleans the tip afterwards. He said this would work well for them. Thank you for that input, Oner. Next question is, can one simply turn on the 405 and 490 light and begin recording without a stabilization period and effectively subtract even the very early and strong period of bleaching?
Yeah, so we see a huge sort of burst of fluorescence when we turn on the LED fibers. So we actually chop off the first about 10 to 20 seconds of every recording in order to normalize the signal. Otherwise, that initial sort of fluorescent burst can skew the normalization. Additionally, it's sometimes initially when you turn on the fibers, you get clipping because the signal is so strong. So just for those 2 reasons, we usually chop off the first 10 seconds.
Okay. Let's see. Then Nolan is asking, how do you flatten the baseline for depiction of changes in signal for consumption bouts over a longer period of times? It has appeared in other examples that there's a good deal of baseline drift.
Baseline drift in the raw signal?
Yes.
Yes. So part of the normalization of the raw signal, so we normalize the 490 or the 470 independent of the 405. And during that normalization, it's detrended, so it sort of flattens that drift in the baseline signal, if that makes sense.
Yeah, I'm not quite sure I fully understand the first part of that question. I'm trying to get to, so there's the raw data, and it shows that there is drift in the signal, and that the rate of drift is a little bit different between the 405 and the 490. That's after we apply the polyfit to each channel, and that is the normal signal.
And he's oh, sorry.
Yeah, so I just don't understand the question well enough to determine how to answer it based on the consumption part of the question. I apologize about that.
Okay, he wrote, yep, thanks. So I believe that that was answered. Let's see. Vivian Nguyen Chi is asking, how do you build the gizmo flow for multiple TTL pulses or stimulation patterns rather than controlling them with an analog stimulus generator? So in the TDT software, I assume that you're controlling a laser digitally. So in the TDT software, it also has, you actually have 24 channels of digital I/O, and so you can use that. There's the Synapse software also has gizmos for generating pulse trains and pulses. And if you would like me to show you, as Patrick had mentioned, there's We have a screen share session, so we can arrange a time where actually I can connect to your computer directly and show you how to set that up. All right. Okay, and then I have a question from Roman. How often every day? I think he means, oh, I'm sorry. Well, and then how long can a session be to avoid excessive bleaching? Can you do several 5-minute sessions on the same day, for example?
Yeah, definitely. So we've kind of played around with it during the drinking sessions where we record for, 10, 15, 30 minutes and then allow the signal to sort of equilibrate again and then record again, 30 minutes later. So I think that's definitely possible to sort of deal with photo bleaching issues.
Okay, great. Yeah, actually, I'm going to go ahead and bring the screen back to me just for a second, just because we had a couple questions that kind of address this. So basically, as Patrick had mentioned, we have these great, I'm going to flip through this. These are kind of some supplementary screen shares that I had. So from our website, we have this screen share tool. So any of these questions for configuration, if you're having any trouble, you can just give us a call and say, hey, I'm having difficulty with this. Can someone connect to my screen? So our phone number there is listed below, as well as the support site where you would actually go to do the connection. But if you just call in, you can be walked through. And that phone number is on our website. I'd also like to mention too that we now have kind of a collection of community examples, as well as we also have SD tools for Matlab toolkits and so forth, which can be found at the address below. And I think that'll help with. I know also a lot of the questions in the registration were asking about how you average sample or bring out a very small signal and a large background noise. So yeah, I just wanted to quickly point that out. Also too, with that, in this SDK too, there are also some examples as far as controlling for automating your controls with Matlab. So you can do things like have Matlab automatically turn your light sources on for 5 minutes and then off and so forth to make these types of modulations easier. Okay, next question from Philip Comers. For analysis and data presentation purposes, what sort of filtering or smoothing are you applying to a signal? Is this different based on the duration of the trace you're looking at, i.e. looking at the 32.5-second traces versus several minute-long oscillations? I'm going to go ahead and switch the screen back to you guys in case you would like to use.
We're actually not applying any filters at all at this point in our analysis.
Oh, okay then.
So those 32.5-second traces are average. So the signal looks smoother because it's averaged across, 50 to 60 bouts across 5 or 6 animals.
Okay. Next question is, okay, I think this was already answered, but I'm just going to go ahead and say it again. Just, I'm wondering what the optimal LED intensity 405, 490 nanometer to get the best delta F over F without photo bleaching. I believe as that was also answered by as Patrick was mentioning for the optimal power. And again, that's something that if you would like to set up a screen share with us, we can help you with. Next question is from Daniel McGee. Did you guys monitor or analyze transients during ethanol drinking behavior?
I'm not sure I understand what he means by transients.
And Daniel, could you provide some clarification?
So we're working through our event detection analysis, and we're not ready to present that yet on what we think are events. But what Jen presented on the transients, or if they're referring to the GCamp signal as transients, these are representative examples of what the signal looks like when the animals are licking, and the licking bouts are shown in orange for ethanol, sucrose, and water. So you can see in all of the cases that when the animal starts licking, the GCamp signal drops. And this is consistent with the published ePhys literature that Jen presented at the beginning of her talk.
Okay, great. All right. Well, I believe we've reached the bottom of our list. I hope, unfortunately, this question especially is a little bit convoluted, so hopefully I didn't skip over anyone. But we have now gone beyond schedule. So I believe I'm going to go ahead and end this here, unless Patrick and Jennifer, is there anything else you'd like to say?
No, just thanks for the opportunity to present our work.
And feel free to contact us if you have any other questions.
Excellent. Thank you both. Yeah, we very much appreciate it. Okay, great. And so just for anyone that came in late, we're going to be, this session was recorded. We'll be posting it on the website later. So if there's any sections you missed, that'll be available for you to peruse. And in the meantime, if you have any questions, please contact Jennifer or Patrick or I. Thank you all for joining us. I hope you got the information that you were looking for. And if not, please send us feedback on, we hope to make this a series of webinars. So if there's anything in particular that you would like to see in future webinars, please let us know. Thank you very much, everyone.